اليوم راح نبتدي بدرس جديد The Synapse and uh, the mode of transmission of the nervous message in a synapse. هلا first, what is a synapse? Here you can observe in this photo how we have a neuron, the first neuron, and here we have another neuron. And the two neurons are connected together by a set of ramifications. يعني the axon that is transmitting the nervous message from the first neuron is ramified into many branches, is branched, and it is related to the cell body or to the dendrites of the other neuron. Here you can observe how a branch of the axon is connected to the cell body of the neuron, the cell body that contains the nucleus. And here you can observe how another part is connected to the dendrite. The connection between the first neuron and the second neuron is called a synapse. And this synapse can be axo, since it is part of the axon, axosomatic synapse. It means that the connection is between the axon and the cell body. It can be an axodendritic synapse if it is connected from the axon to the dendrite. And in some cases, it can be an axonal synapse if it is connected to the axon, either from this site or also from this site, from the end. It can be connected at the end of the axon, and it will be called an axonal. These three types are the types of the synapse between two neurons. Some of the synapses are made between a neuron and a muscular cell, and in that case they will be called neuromuscular synapses or motor endplate. But in this case, all the synapses that we talked about are neuronal synapses. It means that they are the connection between two neurons and not one neuron and an effector cell, as in the case of the neuromuscular synapse. This is first the synapse, its meaning, its location, and its role. It is obvious to make the transmission of the nervous message from one neuron to another or from one neuron to an effector cell. In order to explain more, how is this synapse making conduction of nervous message, we have to start by this part of an exercise. This is an exercise of the 2007 session, the second session of 2007, the exercise of nervous system. In this exercise, you can take a little time to read carefully all the given, and after you finish, you will complete with me. They ask you in part 1 to interpret the results of the first and second experiments. Let's see. In the first experiment, the nerve fiber N1, this is the nerve fiber N1, which is a part of the neuron N1, was stimulated by S1. This is the stimulation S1. An action potential was recorded in R1. We observe here an action potential recorded by this oscilloscope and then in R2. Then in R2. So the action potential is recorded here and then it is recorded here. You can take a meaning from this experiment that the action potential is transmitted from the neuron N1 to the neuron N2. But in the second experiment, the nerve fiber N2, which is this one, is stimulated, where in S2 it records only an action potential in R2. This means that the action potential that is recorded in R2 is not transmitted to the neuron N1. You can say, in other words, that the transmission of the nervous message is unidirectional from N1 to N2. That's why the neuron N1, we will see it after, is called always a presynaptic or presynaptic neuron, and the neuron N2, it will be called postsynaptic. Postsynaptic means after. Presynaptic means before. This is the synapse, and I have a neuron before and a neuron after. This nomination is given because that nervous message transmits in one direction from N1 to N2. We will see after a while that the synapse that is here in this figure more clarified contains, it has a structure that will be explained later, but now I will only note that it contains some substances, chemical substances, and the chemical substances are found only in the presynaptic neuron. These are the chemical substances that are called neurotransmitters, and they have many types. One of the most known types is the acetylcholine, that we will explain some information about it later. Now, for this experiment, here I have the part 2 deduced from the third and fourth experiment, the level of the nerve cell at which acetylcholine acts. What is acetylcholine? We already said that acetylcholine is a substance found here in this site in some vesicles that are called vesicles of neurotransmitters. They are found in the presynaptic neuron, as we said. Now, they said in the third experiment that a microdrop of acetylcholine was deposited at the level of the zone S between N1 and N2. The acetylcholine is deposited here between them, and action potential was recorded only in R2. So this substance, when it is found in the place between the two cells, and this place is called synaptic cleft, they will give you an action potential in the postsynaptic cell. But if in the fourth experiment this microdrop of acetylcholine was injected in neuron N1, inside the neuron, or inside the neuron N2, no action potentials were recorded in R1 or in R2, and this let you conclude that acetylcholine acts at the level of the synaptic cleft. It means between the neurons N1 and N2. This will help us to understand the mode of function of the synapse that is given by this figure from the same exercise of session 2 in the year 2007. The two documents show you the vesicles. These are the vesicles that are containing acetylcholine inside of them, and they ask you to indicate in which state of the two states of documents 2A and 2B the synapse is in function. We can observe here that the vesicles that contain acetylcholine are more close. Some of them are opened to the synaptic cleft, and we can observe the same color of the substance that we observe inside the vesicles. It is between the two neurons in the synaptic cleft. So we can easily find the role of the vesicles that pour or release their substances, the neurotransmitters, in the synaptic cleft, and we can conclude that this is the synapse that is in function, but this one is at rest. And from this we can understand more how does a synapse work. It works by making exits of the neurotransmitters. This is what we call exocytosis. The exocytosis is the release of neurotransmitters in the synaptic cleft. But what do the neurotransmitters make in the synaptic cleft? What do they do? In another official exam, this one is in the 2003, also the second session. This experiment can show you some important ideas. In the first experiment, acetylcholine is injected in the same place, which is the synaptic cleft, and we can record in the postsynaptic neuron some results, some variations in the potential, and we can observe how if we increase the concentration of acetylcholine, the response of the postsynaptic neuron will be greater, will be more important. But the point here is in the second experiment. In the second experiment, they ask to use labeled acetylcholine. The labeled acetylcholine means that the acetylcholine can give radiations. It can be observed because of radioactive rays or radiations that are emerging from it, and we can observe where the radioactivity appear to know where is the acetylcholine going. And we know from the experiment of 2007 that the acetylcholine doesn't go inside the neuron. But where does it go? By means of autoradiography, the plasma membrane of the postsynaptic element appeared radioactive. The plasma membrane of the postsynaptic element appeared radioactive. It means that the radioactivity appears where? In the postsynaptic membrane, here at this place. This will make us know that the acetylcholine, after its release from the presynaptic neuron, it will bind on the membrane of the postsynaptic cell. And you know that every substance that will bind on a membrane has on this membrane some receptors, and the receptors are some molecules that can fix or can bind other molecules to make a function. In this case, the function is to make an a nervous message or to initiate a nervous message in the other cell. This experiment lets us to know that the acetylcholine that is going to go outside the presynaptic neuron will bind on these receptors that were on the postsynaptic neuron. What are these receptors? What are they doing? This new information will be explained from. You can observe in this video how a synapse is what is the structure of the synapse, and you can also observe the vesicles that are inside of it that contain neurotransmitters as acetylcholine. You can observe also some of the functions. The red channels are Ca2 plus channels that allow Ca2 plus to go inside the presynaptic cell after action potential. This will lead to exocytosis of acetylcholine or neurotransmitters that bind on the postsynaptic receptors, the green ones. These are channels, ionic channels that are chemical dependent, not voltage dependent. They will open and let the Na plus go in. The entering of Na plus will lead to some variations in the potential, an increase of the potential that is called EPSP. Now the neurotransmitters will be again deactivated, destroyed, and reuptaken by the presynaptic neuron. It means that they are absorbed. The neurotransmitters are absorbed again by the presynaptic cell. This is called reuptake. So the steps of the synaptic transmissions will be: first, an action potentials arrives. Second, Ca2 plus channels open. Third, the Ca2 plus go in. Ca2 plus leads to exocytosis of the neurotransmitters from the vesicles in the synaptic cleft. Neurotransmitters will bind on the receptors. Then the receptors will open. They are Na plus channels in this case. And the opening of Na plus channels, chemical dependent channels, will make the Na plus go inside the postsynaptic cell. And the entering of Na plus ions in the postsynaptic cell will make a hypopolarization and increase of the value of the negative value of the membrane potential from minus 70 to minus 60 to minus 50. It depends. And after that, the neurotransmitters will be degraded and will be reuptaken by the presynaptic cell in order to be reused in another synaptic transmission. This is the whole procedure of the transmission. And now we can understand why the nervous message is transmitted also in this direction, not in the other one. Since in this side, no vesicles of neurotransmitters are found, and we have no receptors in the presynaptic membrane in order to bind the neurotransmitters and make another new action potential in the presynaptic cell. Next, we will in the next video solve this exercise that is very important. This is the exercise of 2008 first session. That is an important exercise that make you understand more the function of the synapse and practice on some of how can we ask some questions about the functioning of the synapse. Thank you.